In the last chapter, we looked at different cell structures, from cell wall to cell membrane to all the structures inside the cell. In this chapter, we're going to focus on one of the structures, which is the cell membrane, also known as the plasma membrane. The plasma membrane is really an amazing structure because it defines the boundary of the cell, and if this boundary is damaged, then the cell is not going to survive. Okay, so the cell membrane performs many functions. I'm just gonna give you a quick overview, and then we're gonna focus on the specifics. So first of all, the cell membrane will regulate what goes into the cell and what comes out of the cell. And that's a very important aspect because you want certain things, for example, nutrients like glucose, to go into the cell because it provides a fuel, and you want the waste molecules to come out of the cell, right? And for pathogens, like bacteria, virus, you want to keep them out of the cell so that they cannot infect the cell. So all those things are performed by or achieved by the cell membrane. The cell membrane itself is not rigid at all. It can be really, really flexible, and this allows the cell to change shape. Okay, so for example, red blood cells, white blood cells, a lot of times they have to go through very tiny structures, very little space, so they can change their shape so that they can squeeze through. The cell membrane also carries markers for cell recognition. So this is how your body can tell whether a cell is self, okay, coming from your body, or non-self, coming from another individual, another species, from external source. This is also the basis for the rejection reaction when we do like a organ transplant. Okay, so the patient's body can recognize, using the cell markers, that this organ, the cells from this organ are not our self cells, so we need to attack them. The cell membrane also carries receptors for self-signaling and communication, and interestingly, this provides a mechanism for viruses, okay, to trick the cell and get into the cell and cause infection. Okay. So we'll talk about that a little bit more later. Now, just to give you a quick overview on all the structures in the cell membrane or on the surface of the cell membrane. So there is actually a lot going on. So let's look at the backbone first. Now, if you remember from the biomolecule chapter, the backbone is made up of a type of lipid, and specifically that's a phospholipid, and it's a bilayer. Okay, so if you look at this backbone right here, this is a typical structure, right? The polar head with the phosphate group. That's why it's polar. And then two nonpolar fatty acid tails. This is one layer, and then that's another layer. Okay, so that's the phosphate bilayer. That's the backbone for the cell membrane. There are proteins embedded in the cell membrane. So all these are proteins. Okay, so there's some more information on the next slide. So there's peripheral proteins. Now these proteins are usually attached to the surface of the membrane, right? Like a ship tied to a floating dock. Okay? So let's see, these are the peripheral membranes. Okay, so see they're located on the surface, kind of almost like they're anchored to the surface of the membrane. And the second type is transmembrane proteins. Now these proteins are also known as integral proteins because they span through the entire cell membrane. Okay, see they go through, right? They go through, they don't just stay on the surface. So transmembrane, they go through the entire membrane. All right, now the third is the interior protein network. So these proteins are located inside the cell, on the interior of the cell membrane, and they determine the shape of the cell, and they can also anchor things like other proteins. Okay? So usually these proteins are located on the interior of the cell membrane. The next component is cholesterol. Okay, so in this picture, if you look at the yellow molecule, that's cholesterol. Right? Okay, if you remember, cholesterol has that typical structure, like a typical ring structure, right? The adjoining carbon rings. So these are cholesterol. Cholesterol is only present in animal cell membrane. Okay, so you're not going to find that in the plant cell membrane. So animal cells only. Now the presence of cholesterol really kind of increases the kind of flexibility of the cell membrane and makes it kind of like a fluid instead of a very rigid structure. So in some animals, especially those who live in very cold climates, cholesterol acts as almost kind of like antifreeze in the cell membrane. Okay, so it prevents the cell membrane from becoming too rigid. The next one we're going to look at is the cell surface markers. So these cell surface markers contain carbohydrates. If you remember carbohydrates, simple sugars or complex carbohydrates like cellulose or starch. Okay, so those are carbohydrates. In this group, we have glycoproteins and glycolipids. So glycol indicates sugar. So glycoproteins, there are complexes of sugar and proteins. And the glycoproteins, those are molecules that have both lipid component and sugar component. Okay, so they're indicated over here. So this is a glycoprotein. So you have the carbohydrate component and then the protein. And these are glycolipids. So you have the sugar component and then the lipid component. Okay, so these are all the things that you can find in the cell membrane. Okay, now there's one thing that's not in the text, but it's indicated in the picture. So this is the carbohydrates of glycocalyx. So you don't need to worry too much about this. It really just means that you have a lot of sugar, on the surface of the cell membrane. So it makes the cell membrane kind of like a sticky, right? When you think of sugar solution, it can be really sticky, right? And it's the same thing for the exterior of the cell membrane because there's a lot of sugar attached to it, so it can be very, very sticky. So just real quick, we have gone over this information in the previous slide using the animation. So just real quick, point out a few important pieces of information. So remember, the backbone is a bilayer of phospholipids, and you need to know which part is polar, which part is nonpolar. Okay? So it's right here, and then the membrane is studded with proteins. And there are 3 types of proteins that are associated with the cell membrane. Okay, so we went over each of them. And then we have cholesterol, right, but only in animal cell membrane. And then the last one is pretty important. Okay, so we have carbohydrates attached to some of the proteins or lipids in the cell membrane. And usually they are on the outward facing surface of the membrane. So they're facing to the exterior, the extracellular matrix. Okay. And these complexes function as cell markers, right? So we can use that to identify a cell. Okay, so now since the cell membrane is so critical in terms of viral infection, pathogen infection, so we're going to look at how viruses infect specific cells. Okay, now in this picture, it shows the HIV. What does HIV stand for? There you go. So it's the human immunodeficiency virus. So if you're infected by this virus, you are going to have impaired or compromised immune functions. So you're more likely to get infections. Alright, so this is pretty interesting. It talks about the origin of the HIV, the virus. But I want to point out a couple things that are related to the cell membrane. Okay, so this is a human cell. This is a human T cell, because HIV specifically targets the T cells and some of the monocytes. So T cell, it's a type of immune cell. Okay, so that's the target. So this is a T cell, and this is the cell membrane of T cell, and this is the HIV virus. Okay. Now, the proportion of the size is not correct because they want to show you, all the different components. So the virus usually has kind of two main components. The first one is going to be a protein coat, and there may be, carbohydrates, proteins and stuff in this protein coat. It's like a capsule. And the inside, that's the genetic material for the virus. For HIV, it's RNA. It's not DNA. Okay, so it's a one-strand RNA. Now, in order for the virus to infect the cell, to successfully infect a cell, the virus has to find a way to get into the cell. Okay, so in this case, the T cell has CD4 receptor. Remember we said over here that the cell membrane carries different kinds of receptors for signaling and the communication. So in the HIV case, the T cells have this CD4 receptor and a co-receptor. So the HIV has something on the coat that can trick the receptor and the cell into letting the virus into the cell. Okay, so the cell has the binding sites on the surface, that's the CD4 receptor, and the virus can exploit this receptor with specific glycoproteins in their coat. Like they have the components that can kind of activate or trigger this receptor or bind to the receptor. And once that happens, the cell says, "Come on in, you're a friend, come on in." So the virus coat can fuse with the cell membrane, and it's going to inject. The virus is going to inject the RNA, its genetic material, into the cell membrane. And if you remember, this is the nucleus, right? And then everything between the cell membrane and the nucleus is the cytoplasm. So once the RNA reaches the cytoplasm, it's going to use an enzyme to make a DNA. And that's almost like the viral DNA. Not directly, but it's made from the viral RNA. And then the viral DNA can get into the nucleus and kind of incorporate itself into the human DNA. And then whenever the DNA is replicated or transcribed to direct the synthesis of protein, that means your cell is going to make the proteins directed by the viral genetic material. So the cell becomes a little machine for the virus to make more viruses. Okay? So this is how the virus can affect our T cells. Okay, so it's actually a quite interesting process. Okay, so this slide is pretty simple. I know there's a lot of text, but it really just talks about the fluid nature of the membrane. Okay, so again, it's because of that fatty acid tails, hydrophobic, but it can add kind of this fluidity to the cell membrane. And another contributor is the cholesterol. Remember we said that cholesterol makes the cell membrane very flexible, right? It almost acts like an antifreeze. So the fluid mosaic model, it's pretty simple. It's really just kind of talks about in general, all the components in the cell membrane can flow and change position. Okay, very flexible, but they still maintain the basic integrity of the membrane. Okay, so that's pretty much what that fluid mosaic model is about. Right? So they're dynamic and they're constantly in flux. They're not static or rigid. If you're interested, you can look at animation of the fluid mosaic model, and you can see how the cell membrane kind of flows like a wave, right? When you're at a baseball game, you do the wave, it's kind of like that. And then the proteins are kind of little boats in the water, so they can move around. They're not, anchored to any position, right? They can move around, flow kind of almost inside the cell membrane. Okay? We mentioned this a little bit in the biomolecules chapter, but again, because it's so important, I just want to do a quick overview. So this is the structure of the phospholipid. And then this is the polar head. Again, why is it polar? Because it has this phosphate group, which carries a negative charge, right? Anything that carry a charge, whether it's positive or negative, they're going to be polar. They're going to love water. And then this is glycerol component. It's a little bit less important, so you can kind of just move on to the next, the hydrophobic tail. So it's made of fatty acid tails, right? The hydrophobic fatty acid tails do not like water. They fear water. So because of the two different natures of the head and the tail, in an aqueous solution, okay, if you put these phospholipid molecules in there, they are going to automatically arrange themselves where the polar heads are going to point outwards, okay, and so they can come into contact with the water, and then the hydrophobic tails are going to face inward so that they don't have to come into contact with the water. They can avoid water. So you may form a sphere, that's very possible, or in the case of cell membrane, they're going to form a bilayer sheet. Okay? And then, eventually you're going to have a continuous membrane, right, made of these lipid bilayer. Okay, so here are some practice questions. First of all, what chemical property characterizes the interior of the phospholipid bilayer? So what about the interior? So that means it's the fatty acid tail. So what is it? Is it hydrophobic, polar, hydrophilic, or saturated? Right, the correct answer is it's hydrophobic. Select all statements that are true about ions. Now this requires a little bit critical thinking. So the correct answer is A. They interact really well with the polar molecules because ions are atoms that either gain or lose electrons, right? So now they carry charges. They carry negative or positive charges. So they can react very well with other polar molecules. That's not true. They do carry charges. They're repelled by the hydrophobic interior of a lipid bilayer. This is also a correct statement. So the correct answers is A and C. Because they're polar, so they are going to be repelled by the hydrophobic component of the lipid bilayer. Okay. All right.